Hello, everyone. So today, Josh Doria is going to speak to us. He is from the San Antonio area. He did his bachelor's degree at Texas Tech and worked with Calvin Trossel. And he's come down here to work with me as a field technician. And he is also working on his master's degree. And so today he's going to talk about his master's research. So.
All right. Thank you for the introduction, Dr. Howe. As she said, my name is Joshua Doria. I'm a master's student in her lab, and today I'll be discussing the refinement of nitrogen recommendations in Texas cotton. So a little bit of background before we get started. This graph here shows cotton yields over the past 3 decades, all the way back to 1990 in the United States. As you can see, the change in cotton yields has been consistently going up, all the way down from 630 pounds per acre in 1990 up into the 800s and 900s in these most recent years. So with these increases in cotton yields, we know that the nutrient recommendations, some of them have not been reevaluated since that time, since these cotton yields have greatly increased. And these recommendations vary greatly by state. And some of them, like I said, are decades old, and newer research has been showing that deeper nutrients may need to be credited to the plants there. So the objective of my study was to evaluate nitrogen recommendations in Central Texas. The reason we're doing this, like anything else, is in theory to save producers money, be more efficient. Fertilizers can make up to 1/3 of any producer's budget. Most of y'all in the industry have heard about the skyrocketing fertilizer prices recently. This graph demonstrates that. This is retail price of UAN, which is urea ammonium nitrate. It's a fertilizer I use for my study. It's a pretty common one in the field. You can see the red line, which is the 2021 price, jump up quite a bit this past year, going up to $500 per ton. The 5-year average is the dotted line there in the bottom, holding right around $290 per ton. So you can see this is as relevant as ever. Another reason we want to make sure we're applying the correct amount of nutrients is pollution. Many of y'all have heard of increased runoff in the Gulf from farmlands that run across the Mississippi, and extra fertilizers will leach the profile or go into nearby water bodies and can translate into the Gulf and cause issues like eutrophication to the environment. This project is a collaborative effort across the Cotton Belt, so multiple institutions are joining us and participating in this, but I'll just be presenting my research here from Texas, but there are folks doing this all the way up into Mississippi, or excuse me, North Carolina as well. So my experimental design went over 6 years, or 6 site years, excuse me. The 2 years were 2020 and 2021, and we had 2 locations and a few different management systems. It was a randomized complete block design with 4 reps, and the 5 nitrogen rates we had were 0, 45, 90, 135, and 180. The sites we decided to pick were just down the street here at the farm. We have both an irrigated and a dry land study here at the farm. The irrigated is with the linear sprinkler over at the extension fields. That's in the soil series bulk clay, which these high clay content soils that I did my research in are pretty important for the data that comes later. The other site was the Stiles Farm in Thrall. I don't know if y'all have been out there. It's another AgriLife property, and they have limited irrigation ability. So it was a dry land study only, and that was in a Burleson clay soil. So getting through some of the methods and some of the data that we were able to take, we did a lot of soil sampling. The first ones we started with was taking a producer style soil sample and sending it to the state soil testing lab. The recommended when you submit your soil is 6 inches. We also did deeper sampling down to 3 feet, about 1 meter. This picture here is from I think it's from a textbook from 1990. So 30-year-old photo from a textbook showing that we know that cotton roots can reach 1 meter down into the soil by 15 days after planting. And so for those of you who know cotton, that's like 1/3 or 1/4 of the way through the growing season. These roots are all the way down to 1 meter. And we've known this for decades. Some of the other data we looked at was inorganic nitrogen. When I say inorganic nitrogen in this presentation, I'm talking about nitrate and ammonium. These are two potentially available forms that you can find throughout the profile. We looked at it all the way to 1 meter depth where we took our soils. And we took multiple different samplings, both before, during the season, and after. We found these values for the inorganic nitrogen through potassium chloride extraction, and we had to take bulk density samples as well to translate those values from a concentration to a usable stocks and how much nitrogen you have throughout the soil profile. We also looked at lint yield, of course. We got pretty big plots. They're 8 rows wide on 40s here and on 30-inch centers in Thrall. And we sampled 2 rows. They're about 45, 50 feet long plots, so pretty big. And we used a tabletop gin to gin our cotton samples. We also looked at lint quality, which I'll get to later, and plant tissue nitrogen. This was a combustion method looking at total nitrogen inside plant tissue. And the last thing was NDVI, the normalized difference vegetation index, which our last speaker probably knows a lot more about than me. But we did that as well. And I'll be presenting mostly data from 2021 and 2022, or excuse me, 2020 and 2021. But some, we're still getting information we have from this past year together. So it's incomplete from 2021. It'll be mostly 2020 data. So this here is a soil test report you'll get when you submit your samples to the lab down the street. I hope y'all are all completely familiar with this and soil test your soils. If not, I recommend it. But one of the first things I want to call your attention to here is these recommendations are yield-based. So when you submit your soil, you're going to tell them what crop you grow. In this case, it's cotton. And you're going to tell them what your yield goal is. Again, in this case, it's 2 bales. Based on that yield goal, to meet that metric, they're going to give you the different values for each one of the important soil nutrients. Here, we're looking at nitrogen. You can see it's 3 parts per million there. But I want you to notice it's nitrogen only. They don't account for ammonium here. And so they recommend 85 pounds of nitrogen per acre here. That's pretty much lines up with common knowledge. A common thing you might hear from producers, just a rule of thumb, is 50 pounds of nitrogen equals a bale of cotton. So this kind of follows that trend that you might expect. But we wanted to get a little more quantitative with these recommendations. So the recommendations here just tie together all the different ones we got from the lab in our 6 site years. So you have the year, location, system. Again, the yield goal, we had a smaller yield goal in Thrall due to the limited water. And then how much nitrogen they credit us with in our field. And then the recommendations that they suggest you apply to meet your yield goal. So in College Station, both years, about 90. And then in Thrall, both years, it was 50. So we'll get to the yields, which is the fun part. I know there's a lot of data on the screen, but I'll try to break it down. You got the lint in kilograms per hectare on the y-axis and the nitrogen rates on the x-axis. And you have the irrigated in blue, dryland in green, and then Thrall in gray. So for irrigated in 2020, the thin line across the bar is the yield goal that we went for, so 2 bale cotton. And so while most of the treatments hit that, there were no significant differences between the treatments, including the untreated check. That includes between the highest one, which was the 90 treatment, and the lowest one, which was the untreated check. But you can see the error bars is still, there were samples that hit the yield goal. In dryland, if y'all did any research in 2020, 2020 was a weird year. We didn't get much moisture at all, much rain. So the yields are pretty low. And again, there were no significant differences between treatments in 2020. And again, same thing in Thrall. The line is a little lower because our yield goal was 1 bale an acre, but there were no significant differences between treatments once again. In 2021, these yields are a lot higher. We had more normal moisture in 2021, and we outperformed our yield goal, but once again, there were no significant differences between treatments in each year and location. The reason the dryland and irrigated are so similar in yield, just because it was so wet, we didn't have the opportunity to irrigate quite as much as we'd like to. Unfortunately, we lost the Thrall 2021 data due to technical difficulties. I don't know how many people had to migrate computers to the new AgriLife system, but we did, and that happened. It's not ideal. Most times, talk about yield, people want to know a lot of the link quality. We didn't have a lot of differences in link quality from our treatments. So here is just all of our different studies and the p-values for significance within those studies. As you can see, in general, there were no significant differences in link quality. There were a few in those two, but they weren't enough to really change the value of the lint. And these values were achieved by Cotton Incorporated, who funded the project, and we sent them the lint samples, and they conducted HVI, which is they call it high volume instrument color analysis, and produced this data. So after looking at the yield, we had no significant differences, and we wanted to look back at the soil we had and try to figure out why we didn't have differences throughout the profile. So here are some photos of us taking deep soil samples. It's a hydraulic Giddings probe mounted on the back of an F-350. Y'all might have heard stories about that truck as well. Anyway, we have it now. And we use that to take our soil samples. And we even brought Flat Stanley with us one time. So with that data, we were able to figure out how much nitrogen we have throughout the entire soil profile. This data here is College Station in 2021 and Thrall in 2021. The most important thing to notice is the maroon is the ammonium. And so that's what the soil testing lab doesn't account for. And College Station makes up about half of the available nitrogen throughout the profile. And we do know that, again, it goes down to a meter, this nitrogen, but we know that cotton roots have the potential to access. So it's all within the rooting depth. In Thrall, it's even more so. It's closer to 70%. The recommendation for this trial was 95 kilograms of nitrogen per hectare. And we found in the profile 81.3 kilograms of nitrogen throughout the entire rooting zone. In Thrall, it was even more. We had 68.2 kilograms of nitrogen throughout the profile when the recommendation was 50. So more than the recommendation itself. So the most important thing is that little square right there is what the soil testing lab credits you with, whereas the rest of the boxes, kind of symbolize the rest of the nitrogen potentially through the profile. So they're just crediting you with what's in the small blue squares. Some of the other metrics we looked at were plant height. As you can see, for plant height in the irrigated system in 2021, as well as the rest, there were no significant differences. This is probably due to drought stress. There wasn't a lot of water we had. But in 2021, as you can see here, there was significance between the untreated check and the rest of our treatments. So when water is available and the nitrogen's there, these plants were able to grow taller and have more biomass in general compared to more water stressed environments like the year before. And of course, Thrall, where there is no irrigation and less rainfall. Another metric we looked at was NDVI. We flew the drone that apparently can't buy anymore, the DJI. I did not know that. These two photos are just our College Station locations. We did not fly in Thrall, but, and the two photos show the two of the different images you can get from the data from these flights. One of them is for NDVI, that's the red and the green, and that's our 2020 site. And these were taken right around first bloom when they did the flights. And then in 2021, that's on green scale, I think is what's called. And you can see as the treatments are laid out here on the bottom, a general increase in the green from the 0 to the 180. So while I haven't analyzed this data for quantitative values, I expect to see a difference between treatments, whether it's canopy density or NDVI values. Another thing we looked at was plant tissue nitrogen. This again was sampled right around first bloom. This data is just 2020 here. You can see there was significance between the untreated treatment, and the highest treatment with 180 kilograms of nitrogen per hectare when it was irrigated. But in other situations, when there's less water, there were no significant differences between treatments. This last set of graphs is the change in nitrogen in my College Station locations. So this is looking at the amount of nitrogen throughout the profile before we planted, before fertilization, anything like that, and simply subtracting it from the amount of nitrogen throughout the profile after harvest. So as you can see, the general trend is a drawdown in nitrogen across nearly all treatments in all treatments and in all horizons of the soil profile. The one that's not true, where there was an overall increase of nitrogen throughout the profile is what you'd expect. It's the treatment with 180 kilograms of nitrogen per hectare, where there was an increase in the amount of nitrogen in the profile. So we expect that this loss is from plant uptake and some leaching. Again, these soils are pretty high clay content, so we don't think the leaching was going to be too high. But we also think there'd be higher leaching in the irrigated trial. There's more loss there. But we also think there'd be more plant uptake in the irrigated trial. These plants are growing taller and they're greener than the dryland trial. So some of our conclusions, nitrogen recommendations are not translating into yield differences. Results suggest that the current methods may not be accounting for all nitrogen pools throughout the soil profile. And these specifically are available ammonium and deep soil nitrogen. So some of the recommendations we have, first off is a test for ammonium, add that to the soil test. Another thing would be to sample deeper depths. Farmers would balk at the idea of sampling to 36 inches because it's not easy, you need the equipment to do so. But even something like sampling down to 12 inches, something like that, could give you a better representation of how much nitrogen is in the entire profile. And if you consider potential monetary loss from nitrogen application when you're not getting a significant yield difference, then maybe those values would make having someone come out with the probe truck to sample not so bad. A few things we'd also like to do is add soil type to the recommendation form. We don't expect these values to hold up in a sandy soil in West Texas. There'd be a lot more leaching of nitrates there. And so we expect that the residual nitrogen wouldn't be as high, but here with our clay soils, that was a little different. Irrigation also, we saw in 2021, if there was not water present and you're applying nitrogen, it seems to be going to waste. And then past land use. The trials we ran were on land that's been farmed since probably before I was born. And so they've been probably fertilized with nitrogen every year as well. And so the accumulation of nitrogen throughout the profile has been long running. Maybe if you were starting a farm on CPR land in West Texas or something that comes out of vegetative natural habitat and putting it back into farmland, you wouldn't expect to see the amount of nitrogen throughout the soil profile. So I think that's important as well. And the fact is, a better understanding of all of this can help save producers money and also benefit the environment as well. So I want to say thank you to Cotton Incorporated for funding this project and for their guidance throughout the year. I want to say thank you to my community members and Dr. Howe in particular, my chair, and then graduate students and the student workers, some of them are here today watching, and the Cotton Extension Group for a lot of help with field work. So if I have any questions.
Questions? Who's got questions?
So I was wondering, generally speaking, we think of one of the reasons that we don't test for ammonium is because we think that particularly in oxygenated environments, it's very thermodynamically favorable that the ammonium is going to be transformed into nitrate. Do you have any particular even conjecture as to why that doesn't appear to be the case?
Can you shift the question, please?
I'm just wondering if there's a reason that you can come up with why that ammonium is staying as ammonium and not being transformed into nitrate.
I think there could be a few reasons. I mean, you have to have bacteria that are able to transform, and as well, you have to have the oxygen throughout the soil profile that can that has the ability to have that reaction to transform ammonium eventually into nitrite, or excuse me, nitrate. This might occur less lower in the soil profile, especially with the soils we have. They're high clays, and so oxygen wouldn't be as prevalent in those areas, would be my guess.
Follow up to that question, how far, so when you retested the sample for ammonia, was it like the first sample you collected or from after your first field season?
When the tests were ran,
The second test you ran where you showed us that there was a higher percentage of ammonia in your sample,
yeah.
was that collected after you got your data and you realized that there were no differences? Or was it from the previous sample?
We
you
took those
collected?
samples pre-season. Yeah, we took them
So
before.
you retested them?
Yes, yeah.
Can you say what proportion of that ammonia came from fertilizer? Is it mostly from fertilizer addition or mineralization of some organic matter?
Sorry, say it again.
Is it mostly coming from fertilizer, the ammonia you measured?
The ammonium we had in those samples were pre-plant samples. So that was pre-fertilizer addition. Now it could be residual fertilizer from past years. I'm not exactly sure about the past fertilizer use in the site history for these, but that is a potential origin. Yes.
Thank you.
So I may have missed it. Did you specify what variety of cotton you guys were growing? And do you think you'd expect to see something different if you were to plant a more responsive cotton variety?
Yeah, I did not mention it, but it was 440 here and 400 in Thrall, both years, phytogen. Yeah, I think you definitely could potentially see differences depending on the cotton varieties you planted. Yeah.
What's the percent organic matter in your soil?
Slightly higher in Thrall. I think it was a little over 2% in Thrall, and then just under that here in College Station.
So in terms of just average cotton farmers in Texas, like what kind of percent do they usually have on their own farm?
I would say that it varies by region. In West Texas, you're going to have essentially none, or effectively none. Here, these values in the Brazos bottom were probably consistent. It's not like we had a unusual system like a no-till. We had a conventional till system in both locations.
Slide. I wanted to review the first slide that he had, if he could find it. Well, not the internet, yeah.
That's great. He's very literal.
That one. Okay, so this slide struck me with some curiosity because you said it's been steadily increasing. But I would look that there was one increase that occurred between 2003 and 2004, and two relatively flat periods for 20 years on both sides, or 10. Any concept of what happened in 2004 that our yields jumped up? Was that when phytogen got, or Monsanto bought out someone? What occurred?
I did not know. I was 10.
David, that was about the time that the ochre leaf varieties really took over.
Can you repeat?
That was about the time frame when we saw a tremendous number of ochre leaf varieties be adopted by producers.
It just, I mean, it really does look like too flat.
Oh, it was a dramatic genetic difference. And when you were out in the field looking at the difference between some of the older technology stuff and the newer stuff, one would think that there is definitely a difference in nitrogen use efficiency on the newer genetics than there was on the older materials.
The other question I had was, what about CVs? What were your CVs? Coefficient of variation. What was the CV on the experiments for the,
I couldn't say off the top of my head.
for yield? Because sometimes when you show no significant difference in everything, it's because you didn't have very good measurement.
Okay.
You had a lot of variation between reps.
Right.
And sometimes you have really low CVs and you still have no significant difference. So I was just curious as to.
Yeah, I couldn't tell off the top of my head. I'll have to look at it. I remember in Seth, they said that a normal CV would be like 10-ish, I think, but they also gave us metrics for different types of trials might go in different directions. Or might you expect, do you think it's higher or lower for fertility trials? I'm not aware off the top of my head.
Well, just in general, CVs of 10 in cotton or green.
Right.
But for wheat, they're quite common.
Okay.
But our ability to harvest cotton yields and our ability to harvest wheat yields and get the exact number is quite different. So that's why I asked.
Okay.
I have no idea.
So since you've shown that nitrogen does not seem to have an effect, do you have any inclination about what other nutrient might be playing?
Yeah, that was the big thing, is what else is limiting? The question could be, do we reach like the genetic ability of the variety? That's an option. I don't think that's what happened. Definitely in 2020, it was water. I think we saw that pretty clearly in 2021, that what was limiting was water through that year. In 2021, we didn't have any visual symptoms of, potassium or phosphorus deficiencies, but more extensive testing in the soil for the soil samples we took could show that maybe we overlooked some kind of nutrient. The soil testing recommendations do show the other nutrient, so if we were insufficient, we did apply more to make sure we covered all of our bases, but we'd have to look at that.
I have a question on this is Tony Provin out here in the soil testing lab. Why did you choose a 0 to 10 inch sampling depth, or 10 centimeter sampling depth?
Right. Yeah. I myself didn't choose that. Yeah, that was Cotton Incorporated when they were writing up the methods for the preseason sampling. That's the values they gave us.
That's an unusual sampling depth from any of the major cotton states.
Yes, sir. Especially considering y'all's is 6 inches, which would be 15 centimeters.
Yeah.
Yes, sir.
So besides looking at just what's being used, looking at kind of that sustainability side, do you guys have any plans at looking how much is actually you're losing to runoff or leaching, and or how would you do that from the field setting?
Yeah, I'm not aware of any plans to do that. I know the study is continuing after I leave. They're doing another year. The fertilization we do, the method is side dress is put into the ground a few inches. So I don't expect runoff will be huge, at least surface runoff, but I think leaching would be the most interesting to somehow collect the data for how much actually leaches for the profile. When we saw the draw of nitrogen out of the soil compared to plant uptake. I don't, other than isotope testing, not sure how exactly you would do that. But I think if you put a nitrogen isotope inside the system, you can track it better and understand where it's going, whether it's leaching out of the profile or going to the plant.
Has anybody ever measured how much nitrogen a cotton plant actually needs? How much it's going to use, uptake, and store away?
I'm sure that's in the literature. I'm not sure what it is.
The answer is yes, we've done that. The common question is, what is the efficiency of uptake? And that differs on every field and between genetic materials.
Here's that question.
Cool. Why don't you hit the chat? Give the chat a chance.
One of the track questions says, how do you calculate delta N? Oh. That was just the amount of preseason nitrogen throughout the profile minus what was left. So if I can get in here, the negative values were a decrease in it was a decrease in the amount of nitrogen before and then comparing to after. The positive values were an increase from before the season started to after the season. I didn't change it.
So is that output negative input or input negative output?
Repeat, please.
My question is, so for delta N, is that input negative output or output minus input when you calculate the difference in nitrogen?
So it wouldn't be input or output. It's just nitrogen throughout the profile, not any of the inputs. So it's before we put in inputs, what we have throughout the profile, and then the whole growing season, including fertilization, and then the values after we harvest.
Oh, okay, I got it. And my second question was, so if the nitrogen is not contributing to the yield, and so the residual nitrogen is also not that high. So where is that like high amount of nitrogen going actually?
The accumulation?
Yes, so you're adding the nitrogen fertilizer, like the maximum 180 kg per hectare. Where is that added nitrogen actually going? Is it going like to the environment as a nitrous oxide or leaching to the deep profile or something?
Yeah, so there could be a lot of potential fates. And I think the easiest way to be able to track the majority of those would be using isotopes. But yeah, we think there could be some going into the environment with volatilization and stuff like that. But I would guess plant use and leaching would account for most of it.
Thanks.
And we're out of time. So if you have further questions, you can contact me later.